So we're continuing kind of our background information in this chapter where we're getting into enzymes and bioenergetics. Enzymes are going to play a very important role in a lot of the different processes we talk about throughout this course. So we have to get some background information and kind of understand what enzymes are and how they work and what are some things that can affect them. So for this first video, we're going to talk about the different classes of biologically important enzymes. We're going to define some basic terms around enzymes, including reactant, product, substrate, enzyme and activation energy, and then we'll go through a brief explanation on the basic function of how enzymes work. So let's do a little bit of background and some chemistry. Again, I know chemistry, but everything biological is rooted in the rules of chemistry, so we have to talk about it. So we're going to be looking at a lot of chemical reactions in this class, and so we have to remember what a chemical reaction is. It's that when we are breaking or forming bonds, right, within molecules. So for example, when we go through the dehydration, synthesis, and hydrolysis reactions we talked about last time in the last unit, that would be a specific example of a chemical reaction, right? We are forming new bonds or we are breaking old bonds. And with this, we have two sides, right? We can kind of create like an equation. Hopefully you remember the basics of this from your chemistry class. But we have the reactants, sometimes called the substrates. These are the things you start with, right? A lot of times with enzymes, you're going to hear substrate, right? That's usually what we call it in biology. But it's the same thing as a reactant, which is probably the term you've already heard so far. And then the thing you get at the end is the product, right? Sometimes we have reactions that can go back and forth, right? Where the arrow is bidirectional, so it can go from reactants to the products. The products can also revert back to the reactants, right? So sometimes it's a one-way road. Other times we can go back and forth between them. But all of these chemical reactions, they have a certain speed that they can go, right? There is a certain pace that reaction will occur in. And unfortunately for living systems, the natural pace of things, like let's say a complex carbohydrate breaking down into a simple carbohydrate, is not going to be fast enough for us to do anything meaningful with. So we need something to catalyze that reaction, aka speed it up, right? We need to speed up these reactions so things that would take minutes or hours or days or even longer can now happen in the span of seconds, right? We need things to happen very, very quickly. And so we have an entire class of proteins that are called enzymes. Now there are ribozymes, which is RNA. I'm not worried about those right now. I'm going to focus on proteins because that's the majority of our enzymes. So they have all the same rules that any old protein has, right? So they're made of amino acids, right? They have the same primary, secondary, tertiary, quaternary structure. All of that is the same. Enzymes are just a special type of protein that can speed up chemical reactions, right? Enzymes do not become part of the product though. So enzymes just basically serve as a place, right? They serve as an assistant for the chemical reaction to occur, but they're not changed and used in the reaction. And they don't change the nature of the reaction, right? All they do is make it go faster. So the substrate you start with and the products you end with is just the same as if you left those things alone and allowed the reaction to occur naturally. So we're not changing what we get out of it. We're not changing the enzyme. It's just a way to speed things up, right? Think like fast forward, right? It's a fast forward button. You don't change the movie, right? If you fast forward through something, you don't change the outcome. And you certainly don't change, if I'm going to make myself sound old here, you don't change the VHS tape when you fast forward, right? Or you don't change your TV, you don't change your Netflix account, right, when you speed things up. Same kind of thing, right? It's kind of a wonky example, but it might work for you. Something else with enzymes. They usually end with A's, A-S-E. So kinase, lactase, amylase, right? A's, not every enzyme ends in that, but if you see A's, you can already kind of know where you're starting, right? And there's a handful of different types. These are just some of the ones, some that we have seen before, maybe a little bit. Others that we will see over and over again. So let's look at a few. So we have a phosphatase. Phosphatase enzymes remove a phosphate group. So they just strip a phosphate off. Hence the name, right? Phosphatase. Kinase do the opposite. They add the phosphate group. Synthetase will help speed up a dehydration synthesis reaction. So we know what a dehydration synthesis reaction is. A synthetase is going to help that process along. Hydrolase is going to promote hydrolysis. So exact opposite, right? And hydrolase sounds like hydrolysis. Dehydrogenase is going to remove a hydrogen. And then isomerases are going to rearrange things, right? So they're going to rearrange a molecule from one structure to an isomer of that same structure. So we're not adding or subtracting any atoms. We're just moving them around. So going from like something like fructose to glucose. So now I kind of understand what enzymes are, like how do they even work, right? What are they doing? So pretty much like we mentioned earlier, the chemical reactions in the body are too slow on their own, just naturally, so we have to have these enzymes to help speed them up. And the way they're going to do this is they're going to lower the activation energy, right? So basically lower how much energy is required for this reaction to go through. And so think about it, it's like making it cheaper, right? You're lowering the price. You're making something cheaper, and so it's more affordable, more people can buy it, right? So we're lowering the activation energy, we're making it cheaper, right, for this reaction to occur. And so this is going to help increase efficiency as well. So we're going to lower the activation energy, we increase efficiency because the substrates, the reactants can come together, right, on the enzyme. There's usually a binding site, and that way the substrates are coming together so that as soon as they come into contact, they can go in through their reaction, and we're making it easier for them to go through that reaction. And you may be wondering, okay, well, cool, they lower the activation energy, but like, how do they do that? You know, what is the enzyme actually doing? What the enzyme's doing is when those substrates bind, the bonds on those reactants have a very specific setup. And but they're not solid, right? We can twist them a little bit. We can bend them a little bit. So what the enzyme is doing is it's taking these reactants and it's distorting those bonds just a bit, right? Just a bit to make them easier to break, right? So think like if you were to, you had something that was like a piece of plastic, right, that you need to break into two pieces, right? If you twist it, if you start manipulating it, you can weaken it to the point where you can break it. And so kind of similar here, that our enzymes are taking these reactants, distorting the bonds just a bit so that it's easier and takes less activity or less energy to break them. Now we mentioned the enzyme coming together with the substrate. So this is where we come into how this enzyme is only going to work with certain substrates. If an enzyme, let's say we're looking at lactase, right? Lactase is an enzyme that works to break down lactose. Lactase is not going to work on sucrose. It's not going to work on maltose. It's not going to work on any other substrate. So enzymes are very specific to basically one reaction. This is because of the link between structure and function. So if you remember from the last unit, we talked about that when we talked about proteins, that structure and function are fundamentally linked. The structure of the enzyme needs to be exact. It's unique to the reactants it works with. And this is because we're going to create pockets that are called active sites. These active sites are where the reactants can just kind of slip into, kind of sit in, right? Like a key in a lock. And then once they're sitting in that spot, the enzyme can twist them a little bit, distort that bond. Right? But if the active site is not the right shape, the reactant cannot seat itself there. So if something were to happen where the structure of the enzyme were to be changed, it would not be able to work on the reactants it's normally made to work on. So there's a very strong link. And we will be looking at that, right? We will be looking at how we can distort the structure of an enzyme and how that can impact the function of it. So what can distort the structure? It can be things like heat, right? Temperature. It can be pH. It can be solute concentration. There's a list of things, right? The things that would affect a protein's structure will also affect enzyme structure. So, but they're very specific, right? So like the lactase I was just talking about is only going to work on lactose because lactose is going to fit into the active site like a glove. Something like sucrose is not going to fit. And so the enzyme lactase is not going to be able to do anything with sucrose. This usually requires a little bit of ATP because it doesn't make it free, right? It just makes it cheaper. And so we have the substrate, like lactose, coming together on our enzyme's active site, our lactase, a little bit of ATP. We can distort the bonds and then break the thing into two pieces. Now, this whole like distorting the bonds and how the reactants fit in, it's not always going to be an absolute perfect fit, but as the substrate moves into the active site, the enzyme will kind of fit itself around it. This is called induced fit. So it's not like just snap on like a Lego onto another Lego, but more of like, okay, it's gonna get there and then we can manipulate the enzyme around it in what we call induced fit. So our substrate, it's bound to the active site. We're forming a very temporary bond, right? The enzyme then is going to distort those bonds to weaken them and allow them to break more easily. Enzyme weakens the bonds, right? We have this what we call an enzyme substrate complex, which is just the substrate and the enzyme all together. The complex then breaks those bonds or builds those bonds, whatever we need to do, right, to generate the products. And then once we get the products, they get booted out of the enzyme. So it's not too crazy of a process, right? If you can look at this picture that I have here on the right and give a brief explanation on how this works, I would generally be pretty dang happy, right? That would be something that you should be able to explain, like what an enzyme is and the basics of how it works, whether that's through words or through a diagram.